Going to start chapter 2. Did any of you, anybody? No, right? Had you read ahead, well, some of your classmates did. They're not here today. That's pretty exciting, right? And the reason they're not here is because chapter 2 is quite possibly the most difficult chapter in the entire book. Okay? That in chapter 4, 6, 7, 8's pretty tough. Chapter 2 is the most difficult chapter. The reason it's the most difficult chapter is for those of you who have not had physiology. Anybody have a basic physiology course? Okay, so you guys are going to be fine. The rest of you, I'm really sorry. Just hang in there, right? So this chapter is going to be tough. We're going to have to really talk about structure and function, right, of the cells, right? So we're thinking about the anatomy and the physiology of these cells. We're going to really get down to the details. Probably not today. I'm probably going to just stop right at the point, or at least not go through the full sort of meat of what's going on. We're going to get right up to that point. Maybe we're going to stop, give your brains time to relax a little bit, and then keep going forward. But this is going to be the most difficult part of the entire course, because you're going to have to start thinking about ion channels and movements of ions, right? So it's going to be a different kind of thinking than maybe you've done in the past. All right. We'll talk about the cells of the nervous system. We'll talk about communication within a neuron and between a neuron. It's going to be exciting. All right. So you've got a nervous system. It's there. We can divide the nervous system into different parts, right? And there are different ways that we can and will divide this nervous system and categorize pieces of it as we go through. Sort of the first broad set of categories that we have is what's called the central nervous system, or CNS, and the peripheral nervous system, the PNS. Okay? What is the central nervous system? It's only two things. Two things only. That's your brain and your spinal cord. It is nothing else. Okay? Brain and spinal cord, that's it. All right? What's cool about your brain and spinal cord? They're both encased in bone. Got a skull, got a vertebral column, right? They also have some nice, tough wrapping on them. We'll talk about what those are. Anything else that's not brain or spinal cord is going to be out in the peripheral nervous system. This is going to include nerves that are attached to the brain or spinal cord, as well as anything else going out through the rest of your body. Okay? And in case you didn't understand any of those words that I said, we're going to put it up here just in a nice picture for you. Brain and spinal cord right there in red. Everything else is in blue. That's peripheral nervous system. All right, does that make sense to everybody? Okay. Your nervous system really does three things, and three things only. Okay? It's a three-step process. We can take this three-step process that I'm going to give you, and we can apply that to the entire organism. We can bring that down and apply that to an individual cell, to a circuit, a group of cells, even all the way down to ion channels. We can really drill down with this, right? And it's three steps. Input, calculation, and output. That's it. That's all there is to it, right? Your entire nervous system is collecting inputs. It's taking those inputs and calculating something, right? It's putting that information together. It's making sense out of that one way or the other, and then it's creating some kind of output. Now, in this sort of example, we have a sensory neuron, so that's going to collect information. Look at that. Nice stare down at a glass of water, right? I'm sure you all stare at water that way sometimes, like, water, I'm definitely going to drink you, just watch out. That's that intense stare, right? This guy was probably trained at Juilliard or something to stare at water that way. Next, we have the interneuron. The interneuron is where we're doing some sort of calculation, right? We got, hey, we see a glass of water. That's awesome. what are we going to do about that glass of water, right? What are the things that those inputs are doing for us? Finally, we're going to have the output. The output in this case is going to be a signal to a motor neuron. Reach out, grab that glass of water, right? Input, calculation, output. It's pretty simple. No. It's a good question. Well, I mean, sort of. I mean, I guess we're there. If you want to be, it's up to you. So when we're thinking about, right, afferent and efferent. Okay, what do we mean by that? We're basically talking about things that are coming toward you or toward a target and things that are going away, right? And so in this example, we could sort of think about that being as the afferent information, the information sort of coming in, and then this information being the efferent, what's exiting, right? That's all going to be relative depending on where you are, right? This actual circuit is too simplified, and there are a bunch of steps right here in the middle, okay? And we could have afferents and efferents within that circuit as well, right? So that's kind of an important thing to keep your heads around. That's a good question. How many of you have ever seen one of these? Great. Okay. We're going to talk about a few things here, a few things that's going on. I can never tell if that's the projector or the screen that's vibrating. I don't trust the installation of either. Can you guys see that vibrating? Okay, great. I mean, it's not great that, you know what I mean. It's great, it's not just me. When we think about a neuron, we want to think about it as having a special job, right? It's only really doing one thing. Its job is to collect a signal, make sure that signal travels to its destination, right? That's all it's doing. It's moving a signal from one place to another. We'll talk about the nature of that signal later. We'll talk about the nature of how that signal is created in some detail, right? We're going to kind of build on this, right, Cody? So just hang in there. If all this is like, hey, I have no idea what's going on, if it looks like we're starting in the middle of the story, it's because we are. It's worse now. Yeah, now everybody notices it, right? Just move your head up and down at the right frequency. You'll be okay. So we're going to start in the middle of a story, but we'll keep coming back around to it, okay? So just hang in there, and we'll talk about all this. This particular neuron is a very stylized neuron, right? It's maybe you have one that looks like this, maybe you don't, right? How many of you have ever seen that fun? Those of you that took like cell biology, probably whatever class you took that involved, thinking about cells, they drew that fun little cell like this, right? You're like, oh, there's a nucleus, and it's got little things coming off of it, and a tail, and all these little parts in the middle, and you're like, oh, yeah, those are what cells look like, right? You don't have a single cell in your body that probably looks like that, right? They put all of the cool pieces into one cell just to give you an idea of what that cell is like. And we've sort of done something similar here with this neuron. This is sort of a standard neuron, right? Later, and in some classes, you might spend a lot of time thinking about bipolar or multipolar or unipolar neurons, right? Anybody ever heard of those things? Yeah, they're kind of cool, right? They have interesting things about them. I'm not going to expect you to memorize all of that. What I want you to take away from that story is, well, if a neuron's job is to collect this kind of signal, then it's going to have a certain shape to do that, right? If it's to collect a different kind of signal, it's going to have a different shape, right? Kind of depends on what specific signal it's collecting and where it's sending that signal, so things might change. Later, we're going to talk about sensory systems in particular. A lot of the nervous system cells involved in sensory systems are very specialized, right? If you think about the cells in your retina, if you think about the inner hair cells in your cochlea, those are very unique to that situation because they have very specialized roles, right? They're picking up a certain signal. But there are some basic commonalities that we can talk about, some things we can think about here, right? First, let's start with your dendrites. Okay, so what is the job of a dendrite? Dendrite actually is from the Greek word for tree, right? Sometimes you'll hear dendritic arbor, right? So it's kind of still a tree word, right? You're thinking about that. So you have your dendrites. This is the main receiving apparatus on your neuron. Okay? These branch out. Kind of depends on the location of the cell, right? You have many dendrites. Each of these little spots is somewhere that it's receiving a signal from some other neuron. We'll talk about those connection points in detail later, right? But it's going to collect all these signals. Most of your, well, on average, your neurons have about 10,000 inputs, okay? About 10,000 contacts. Now, there are some neurons that only receive one contact. There are some that receive 100,000 contacts, right? It's just the average is around 10,000. Why would we have more contacts or fewer contacts, right? What does that do? It changes the power of that incoming signal, right? And there are some times where you only want that one-to-one relationship, right? It's very, very important what that other cell thinks, what information that one cell has. Sometimes it doesn't matter as much, and you're more interested in those broad data collection process, right? And so you can think about this in your own life, right? How many of you live with another person? Now, that happens, right? That might be a situation where what that one person thinks is really important to you, right? Okay, because you have to deal with that person day to day, right? You've got important things going on. When you move out of that and you're thinking about maybe some other choices in your life, maybe it's more important what a broad number of individuals think, right? How many of you have made you don't have to tell me, made a large purchase recently? Anybody purchased, something expensive, right? It's just something where you looked at reviews, you looked at data, you looked at information, right? If you're buying a car, right, you don't just, maybe you do just ask one person what they think, but hopefully you've done a little more research than that, right? You kind of look at what are the reviews for this, what are the experts saying. You look at a lot of data streams, right, a lot of data sources, okay? Most of your neurons are going to do that. Most of the time you're going to collect a lot of data from different sources. Each of those sources can have some sort of influence on your decision-making process, right? Just like some of these contacts can be more powerful than others, okay? Some data sources can be more important in your decision-making process. We collect all of these signals in our dendrites. We send all of these signals sort of down toward the cell body or soma, right? What's in the soma? All of the sort of mechanics that you need to make this cell operate are going to be located in the cell body, right? That's where the nucleus is going to be. We're going to have mitochondria. We're going to have a lot of other things there. There are certain things we're not going to have. There are certain limitations, things that neurons can't do, okay? One big thing we're going to talk about that neurons can't do is they can't store up their own energy, okay? So they're not going to be storing a lot of energy, right? We've gotten rid of that. We decided instead we want to be able to take a signal, send that signal quickly to its destination, and make certain that that signal when it arrives is exactly as it was when it originated, right? We want that to be the same, okay? Other contacts. So you can skip ahead a little bit here. At the end you have these terminals or terminal buttons, right? One of these will actually kind of wrap back around and contact some other cell, right? Some other dendrite. And there are normally, on average, 10,000 of these on any given cell. Some have more, some have less, kind of depends. Purkinje cells in your cerebellum have about 100,000 inputs. Your retinal cells, cells in your early visual pathway have about one contact, okay? But if you pluck out a cell, on average, it's going to have 10,000. All right, so we've collected all this information. We've got it all down here. It's all an electrical signal, right? So what we're doing is we're opening and closing ion channels. Okay? This is a little preview for what we're going to talk about later. That's how these contacts work, right? We're going to open and we're going to close ion channels. How do we do that? We typically do that with things called neurotransmitters. Okay? I mean, you guys have seen those great commercials, right, for different kinds of drugs, and they have the little little things that float across and make the other cell happy, right? That's a neurotransmitter. Does it work exactly like that? Well, no, but we'll talk about those details, right? But if we open and we close ion channels, we can allow positive or negative ions to come in and out. Okay, don't worry about that too much right now. We'll talk about that in detail in a bit. If we get enough positive ions to come in, we're like, oh, that's great. Then we're going to send an action potential down through the cell and down through this axon. Okay? We'll send that signal down. But we have to get all those inputs. Now, from time to time, you're getting inputs that say fire signal. Sometimes you're getting ones that say don't fire signal. Things are kind of going up and down all the time. You're always making these adjustments. Okay? Let's imagine we get to the point where we're going to send an action potential down the axon, right? Most axons, for our purposes, we're going to think about are going to be wrapped in these things called myelin sheaths, right? So there's a set of other cells other than these neurons that make this myelin sheath. We'll talk about those in a moment. They reach out, they wrap around the axon, they insulate that axon, right? And that does a few things for us. It allows us to fire a signal faster. That's the first thing, right? You want that signal to go fast. And two, it allows us to make sure that signal arrives without any sort of alteration of that signal, right? And so it's like a high fidelity, high speed connection. Okay. And that's important. It's very important to our survival, right? Right, Caitlin? Have you ever been chased by a bear? No, well. But you want your action potentials and these signals to go at least as fast as a bear can run, right? That's what I always think about, right? If you're thinking about your survival, you want these calculations to occur quickly. You want to make these decisions quickly. You want to be able to send signals for output quickly, right? Can you imagine what would happen if it took 5 minutes for you to make I'm not talking about some, like decision where you're actually thinking through a process. I'm talking about 5 minutes from the time your motor cortex says, all right, legs start moving, until your legs actually start moving, right? By the time, if you had to wait 5 minutes, by the time it's time for you to move your legs, by the time you actually move them, whatever reason you are moving your legs for has already changed, right? It's already moved to something else, and maybe instead you need to move in a different direction, or maybe you've already been eaten. So we want this signal to go quickly, and it needs to be accurate when it arrives. We do this with the myelin sheath. In between, we have these little gaps, right? These are called nodes of Ranvier. Please do not pronounce it Ranvier. Anybody heard that one? Great. You're too embarrassed to raise your hands. Somebody has, I can tell. Yeah, right. It's Ranvier. He was a French Canadian. That's how you get the pronunciation, right? So nodes of Ranvier, right? These little gaps in the myelin sheath are awesome. We'll talk about it later. They give you a little turbo boost, right, as the signal's coming through. They give a boost to that signal. Again, increases the speed of conduction there, and also allows us to make sure we have a great signal when it reaches the end, right? You do have some axons that are not myelinated in your nervous system, right? So why would you not myelinate an axon? Well, the better question is, why would you myelinate? Because this is effort, right? Okay, this is a lot of effort. You're putting some resources into myelin, right? Because you have a bunch of cells that make up that myelin. If you can get by without it, you'd rather do that, right? Because it's easier for you. Okay, you can save those resources and do something else with it, right? So there are some cases where you would not necessarily need that myelin. You wouldn't have that myelin. We're not really going to think about those that much, so nothing to worry about. Once that signal makes it down through the axon to the end, to these terminal buttons, at that terminal button there's a sequence of actions that occur, okay? The end result of those actions is the release of neurotransmitter, right? The end result is we're going to dump out dopamine, serotonin, acetylcholine, whatever neurotransmitter that is, right? And those are glutamate, GABA, those are sort of the big guys you're going to think about in this class. So we're going to release that neurotransmitter. That neurotransmitter release is going to do something awesome. It's going to open or close an ion channel on another cell, right? And that's going to cause that cell to calculate that information and decide whether or not it's going to send another action potential. And it just keeps going in a loop like that, right? Now, at some point you're going to reach a gap, in this process where the output is not going to be to another cell. It might be to something like muscles. Okay? So that output might be to muscles. In that case, you're going to have some sort of movement, right? And that's sort of the end of that, that situation. Questions about that? We will, I promise, come back and talk about a lot of these. It's a lot right on one slide. We're going to talk about each of these components, and we're going to think about those things. All right. Here is the basic structure of the nervous system. These are the basic parts of a neuron, right? This is a slightly more complicated view showing that same neuron that we just looked at sort of plugged into a circuit, right? And here what you can see are the inputs coming in. And again, we've drastically simplified this. Anybody want to count those contacts? It's less than 10,000. So just trust me on that, right? So we've drastically simplified this. And here is that cell contacting other cells. There are all kinds of weird arrangements, right, where you can reach back and contact some cell that was contacting you to give it feedback about what it's doing, right, which is a great thing. You guys think about that, right? There are a lot of interesting sort of arrangements here. One of the other things that's sort of not to scale in this particular drawing is the axon. Relative to the rest of the parts here, the axon's typically a bit longer than this, right? So kind of think about like a really long sort of spaghetti noodle, right? With like a marshmallow at the end or something. So it's got to be longer, it's going to be thinner. Inside of this axon are going to be microtubules. How many of you love microtubules? I did hear a lot of woohoos. I thought I would. I thought people would get really excited about microtubules. They're cool for a couple of reasons. They will give us structure for the axon, which is great. They also provide us sort of a railway system, right, to move things back and forth. We'll talk about that just a little bit. We'll talk about this this transport, right? So you have what's called anterograde and retrograde. Okay. By anterograde, we think about things going forward, right? So that's going to go from the cell body down to the terminals. Okay. Let's say we produce a neurotransmitter in the cell body. We need to get that down to the terminals so we can dump that out, right, Garrett? So we're just going to send it right down. That's going to be anterograde. Let's say you've got something down at the terminal buttons and it's broken. You got a broken receptor. You got a broken, reuptake mechanism. What are you going to do? Pluck it back up, send it up to the cell body for recycling, right? So that's going to be retrograde. That's going to go in the other direction. Right, Kyle? How close is this to how Ilsen teaches? I need to... So as a it's like an inside to that joke, right? Kyle and I took physio from the same, completely different times. Kyle being quite a bit older than I am. So we took physio. We took this class from the same person at another institution some time ago, right, Kyle? Like what, a year ago? We were both there. I think that's when it was. So yeah, it wasn't that bad. The person who taught that, she had a habit of covering her mouth and then talking while she was inhaling, which I don't know if it's not a great way to get knowledge out there in the world, right? Like, cover your mouth and talk while you're inhaling. It's a real talent. So hopefully the people in the back can hear me the entire time. All right. Questions about movement? I think we're good there. What am I going to ask you about the inside of a neuron, right? The inside of the cell body? I don't know. There's some important stuff here. We are going to talk about the nucleus a little bit. What's fun stuff that happens at your nucleus? There's like something really, really important there. It's so important, we've made it the shortest, like, sort of word possible. It's not really a word, right? You've got like DNA in there, right? And over the course of this semester, we might talk about DNA. We're not going to get into the big D, like, guys think about DNA replication and so forth, right? But we want to think about the products of that DNA from time to time. We're going to think about different proteins, different, different types of proteins, right? Different enzymes. We're going to think about different ion channels, different receptors, different structures. And we'll think about some of this in this class in terms of how these genes vary from person to person, and particularly how they vary from person to person when we think about particular like neurological disorders, right? So we'll talk a little bit about DNA when we talk about Huntington's disease, Parkinson's disease. We'll talk about a few other things as well as we go. So that you kind of need to be aware of. What about mitochondria? How many of you love mitochondria? There we go. I was waiting on the loser to say it. No, I'm just kidding. Somebody's got to say it. You're required, right? Someone over the course of your life has continued to yell at you that mitochondria are the powerhouse of the cell, right? And that seems to, A, be accurate, but B, also like make you not think mitochondria are all that cool, because they actually really are. Mitochondria have their own kind of DNA, which is pretty exciting, right? So it's like maybe different than the other, I don't know. It's like really fascinating. So mitochondria, we definitely need a lot of those, okay? How many of you have a brain? There we go. After the first exam, Kyle, we'll see if that was accurate. Remember that time, see now I'm already starting, right? Remember that time I told you your brain takes up, what, 2.3% of your body weight, right? Okay. At any given moment, a good 25% of your blood supply is headed to your brain. Now that seems like a lot of blood for something that's not that much of your body weight, right? All things being equal, you would imagine whatever part's bigger is going to get more blood at any given time, right? That makes sense. But your brain's actually going to demand more resources than other parts of your body. Your brain is it's an energy sink, right? Constantly doing things. Even as we'll learn in a few weeks, while you're asleep, your brain is doing a bunch of stuff, right? Constantly working. Those neurons are constantly going, right? So let's go ahead and just bust a myth, Kyle. I love the one, you only use 10% of your brain. That's my favorite. There are people who only use 10% of their brain. They're dead. You need a good bit more than 10% to like keep breathing, okay? So you're using all of your brain. Now that doesn't mean all of your cells are firing at one time. That's called a seizure. That's a different thing, right? And so we got to be somewhere between dead and seizure, right? That's what we're shooting for most of the time, okay? Right? That's good. That's my advice to life. Keep your brain somewhere between dead and seizure level activity, and you'll be fine, okay? Makes sense. So you're using all of the parts of your brain, right? Who remembers that guy? Remember Chuck D? We talked about Chuck D, right? And his basic idea was, you know, if you've got it, it's got some function, right? If it didn't have a function, you'd have gotten rid of it and made something that did. Right? So your brain, all the parts of the brain, all the different regions of the brain, it's doing something important for you. If it didn't, you'd get rid of it. Or you'd have never bothered to develop it in the first place, and you'd still have a lizard brain. Right? Why do lizards not have a frontal cortex? Because they don't use calculators. They don't need to do that, right, Sydney? They figured out other ways to solve their problems. Okay? Questions about that? Comments, concerns? Great. That sounded like you dislocated a joint. Yeah, that was a good one. Protein synthesis, we do want to think about this ever so briefly, right? This is related to the DNA that you have, your ability to read that DNA, create a protein. Why is protein synthesis important to us? What are things that you can think of that are proteins? Well, like remember all that stuff I listed, right? Enzymes, neurotransmitters, reuptake, receptors, autoreceptors, all the stuff we're going to talk about that's important, it's going to be proteins, okay? I'm not going to ask you to memorize amino acids, right? Anybody have to do that? Memorize all those codons, right? That's so much fun, right? Yeah. You know when you're going to use those again? Yeah, never, right? They're in a chart. You can look them up. So there you go. All right. That was sort of a story about neurons, okay? There are other cells in your brain, in your nervous system, that we need to think about besides neurons, right? Neurons might be doing the heavy lifting. They might be the people who are like really flashy and it's exciting to think about them, right? The folks who typically have the spotlight on them. But there are a bunch of other cells that are extremely important to the function of your nervous system, right? Collectively, we refer to these as glia. This word means glue. Originally, this word glue, right, glia, was sort of applied in a very literal sense to these cells. It was thought that these cells physically held the brain together, and that was their only job, right, to actually hold that structure together. In fact, they're more of a metaphorical glue, right? They're not necessarily physically holding things together, but they are providing valuable services, things that the neurons cannot do on their own, right? It's important. So, astrocytes are on that list. Why are they called astrocytes? They're shaped like stars. They do some very important things with energy delivery. We'll think about that in a moment. Oligodendrocytes are important for the myelin sheath. We'll talk about those guys in detail as well. And you also have microglia. How many of you love brain infections? You do? It's exciting, right? I mean, it's a lot of fun. Microglia are part of the immune system in the brain, right? Okay, so they're going to help fight off infections, invaders in your brain. All right, let's think about astrocytes just a bit. So, how many of you think your brain's pretty important? Yeah, it's so important, like we have whole classes on brains, right? It's also so important, we wrapped it in some thick bone, right? Right? How many of you, you got a skull, right? You got something important in there. On top of that, so we think about, like, how are you going to get something into your brain? Well, you think about it from the outside, right? So we got that covered, literally, right? Skull, we're ready to roll. But what about internally, right? How can things get into your brain? Well, how do things move through your body typically, right? Like, what's the number one way? If you have something in one location of your body and you want to get that somewhere else, how do you do that? Well, you've got these great things called blood vessels, right? You guys ever heard of these? Okay, they're kind of important. And they deliver stuff, right? It's awesome. They deliver oxygen. They pick up trash, right? So they'll pick up carbon dioxide, dump that off somewhere else so you can get rid of it. They do a really, really important job, okay? The problem is there's not a real control mechanism on that, right? It's kind of a leaky system. Whatever you dump into your blood, it's going to fall out sort of other places, and there's not a checks and balances system there, right? There's not something saying, no, you can't jump out here, right? If whatever's in your blood supply is of the right size and the right, polarity or the right charge, it can easily diffuse out, right? That's how this works. And for most places, that seems to be acceptable, right? That seems to be okay. How many of you, you think about some of the organs you have, if one of them gets damaged, what can you do? You can sort of get replacements sometimes, right? It's not fun, it's not easy. I'm not saying go out there and sell your kidneys because you can get another one, but there are things we can do to get you by, right? If there's a problem with your brain, anybody heard of a brain replacement? Yeah, it doesn't really happen, right? It's not a thing, okay? If you have damage to your nervous tissue, especially your central nervous tissue, there's not a replacement for that. There's not a way to really fix that right now, right? If there's a problem with one of your kidneys, well, the other one will still get you by, right? You can do heart transplants, liver transplants, right? You can even get just part of somebody's liver and let it grow into a full liver, right? And you're okay. I'm not going to say you're great, just saying you're okay. But if part of your brain disappears, stops working, well, whatever that part of your brain did, you're not doing it anymore, okay? Does that make sense? Huh? Kids are a slightly different story because they're weird. And I put kids in the same category with magic people and wizards, and I avoid them as much as possible. But yeah, no, we'll talk about synaptic pruning and stuff in a bit. And that is, you're absolutely right. And there actually are instances too where folks, there's a good bit of plasticity if like let's say you're folks who are born congenitally blind will use their like their auditory cortex will sort of take over their visual cortex. And there are some things like that that we'll talk about, absolutely. So, but because our brain is so delicate, right, and because it's so important, not only do we have this external sort of protection, we're going to have a little bit of internal protection as well, and that's going to be with the blood-brain barrier. Okay? And what is the blood-brain barrier? It's very simple. It is a barrier between the blood and the brain. That's it, right? Now, it's not a sort of like one single thing, right? There are differences we'll talk about in a moment in the capillaries in your brain versus the capillaries in the rest of your body, right? And those differences prevent sort of the free movement of molecules from the blood into the brain. Now, there is a major, major, major exception to this, okay? And right now, I want everyone's attention right now because I'm going to tell you something that is going to forever change your life. This is probably the most important concept. That's an intense stare. I appreciate that. Thank you. This is an important concept to understanding like how life works, right? It's lipid solubility. Okay? Why is this important? We have spent so much time, effort, energy creating this blood-brain barrier so stuff won't get through. How many of you, and you don't have to raise your hands, but we'll know, today's Thursday, right? Is Thursday still like, it's the day, right? It's still the day. So we'll know later today, right? How many of you have ever ingested a chemical that affected your brain function? Yeah, right? What's the number one thing on the top of the list, right? It's alcohol. Alcohol is one of the, yeah, you were thinking of something else, right? But I'd like to use alcohol for this example, right? Because that's something that in this state some of you can obtain legally, right? There are other things we'll talk about later in some detail as well, which will be exciting, right? Right, Kyle? Yeah, they're ready for the LSD lecture. That's going to be fun. Cocaine. Yeah. Tell them about the time I gave rats ecstasy. Caitlin, I was teaching a psychopharmacology class a year or so ago, and I wasn't really thinking about things, and I said to my students, you know, back when I was buying drugs, and then I meant back when I was buying drugs from scientific supply companies under an approved, you know, set of protocols for the research I was doing, there were different things you had to go through. So that's right, Kyle. Lipid solubility. Things, so what are you made of mostly? Right? It's just a bunch of lipids. If you think about your cell membranes, what are those? That's the phospholipid bilayer, right? How many of you have heard of that thing? Right? Yeah, and like stuff moves around, it's a lot of fun, right? Anything that's lipid soluble will slip right through, right between those phospholipids, right? Just like they're not even there. So what's typically getting through there? Like small things, right? Things that don't have a charge, like little oxygen molecules can get in and out. We'll talk about nitrous oxide one of these days, how it's a great signaling molecule in your nervous system for feedback. One of the other things that's very lipid soluble is alcohol, right? Alcohol, when you ingest it very quickly, can get through your cell membranes. It can very quickly get through this blood-brain barrier, right? Slides right through, and it does awesome stuff for you, right? Drastically increases your athletic prowess, right? It's exactly it's what everyone thinks, right? Right, Cody? Not you, but you've seen people, and they've like, oh, look, I've had 2 beers, I can definitely, use this pommel horse. It's not going to happen. Trust me. I saw it on YouTube. So, blood-brain barrier, lipid solubility. What are other things that are lipid soluble, right, that affect brain function? How many of you have heard of SSRIs? Right? Selective serotonin reuptake inhibitors. These are drugs. These are drugs that are prescribed for a number of issues, right? Depression, irritability, a number of things, right? These get into your brain, and they affect, and we'll talk in a couple weeks, specifically how this works. It prevents you from sucking up all the serotonin you release, so there's more serotonin available at the synapse for you to use, right? If these drugs were not lipid soluble, guess what would happen? Nothing, right? You would take the drug, it would never get into your brain to make any difference in brain function, and you would just continue to take this drug and you would never feel any different, right? Drugs that are what we call psychoactive medications, right, that affect brain function have to get past the blood-brain barrier. What's the best way to do that? Make them lipid soluble. They'll slide right in, they can do what they need to do, and you're good to go. I'm trying to think of a like exception to that rule, but I don't have one. So yeah, there's only one other way to get something in your brain, and we'll talk about this because there are times where you might have to do an injection into your brain itself, right? And so typically those are going to be things that are, if you've had some kind of severe brain infection, you know, and we're trying to deliver some antibiotics or something to your brain. But in terms of any medication you're going to take, any drug you're going to take that is going to affect brain function, it's going to slide through that blood-brain barrier, and it's going to have to be lipid soluble. Yeah. Kyle, you got any exceptions to that? Okay. I think that's it, right? Because you don't have, and once you get into your brain, you do have you know, receptors and other things, but to get past that blood-brain barrier, there's not special channels for particular drugs, right? They all just have to slide between the cells, or through the cells, the membranes themselves. You got an example now?
No.
Okay.
That's right.
Specifically, we said that neurons cannot store their own energy, right? And so one job of these astrocytes is to pull glucose out of the blood supply and then dump that in to the brain, to the neuron themselves, so they can use that glucose. Please, I'm going to say a word, and I know it's, you know, we always worry about these things that we might say in class. They're going to cause students problems. I'm going to say Krebs cycle, and I hope that doesn't like send anybody into a tailspin, right? Because that's a rough one. Do you guys have to memorize the Krebs cycle? Yeah, it's like this. No? Nobody? No? You don't remember that? Yeah, right. It was rough, right? Yeah, it happened. If you don't know about the Krebs cycle, just don't bother. You've made it this far in your life, you're fine. I promise. Right, Kyle? Great. You're doing such a great job just agreeing with me every time. I like that. Yeah. Your brain runs exclusively on glucose, right? So when you have other forms of energy, you're going to need to, you know, like long-term storage, we'll talk about that later. We will talk about ingestive behavior and so forth. You're going to need to convert that to glucose so your brain can use it. How many of you love Fun Dip? Yeah, right? Why do you love Fun Dip? It's sweet, right? It's sugary. It's a lot of glucose. Your brain's running on glucose. That's awesome, right? How many of you love cavities and diabetes? Nobody, right? Nobody at all. So we're kind of like riding the line between, like, let's eat as much Fun Dip as we can, but not so much that we lose our teeth and develop health problems, right? This wasn't such a big problem when like the sweetest thing you could get your hands on was like an apple. Right? That's not so bad, right? Because you can go like, hey, apple, I can eat apples all day long. I mean, you're going to be doing something else all day long later, but you can eat apples all day long for a little while, right? And then you're going to, you know what happens if you eat a lot of apples? The doctors stay away. A lot of people stay away for different reasons, right, Kyle? So because we live in a slightly different artificial environment, we can create some problems based on what's available to us. That's a little bit of a preview for some future lectures. Hey, how about oligodendrocyte? How many of you have a dog, you're looking for a name? Oligodendrocyte seems like a good one, right? Nobody's going to name their dog Oligodendrocyte. Ogie for short. I don't know. Ogie, you like that, right? Just write that down. That was a good one. So, oligodendrocytes, what do these guys do? Well, these guys are kind of like squids. They reach out to axons and they wrap around those axons and create small segments of the myelin sheath, right? Now, here in a few moments, we're going to talk about something called the Schwann cell. Okay? At this moment in your life, I'm going to tell you a Schwann cell and an oligodendrocyte are really doing the same thing. Here in about 10 minutes, I'm going to tell you they're doing different things. So just live in that world for a moment where they're the same, right? And their job is just to reach out and they create segments of that myelin sheath. There are some important differences between oligodendrocytes and Schwann cells, right? First, oligodendrocytes are located in the central nervous system. Where do you think Schwann cells are located? Yeah, there's only one other place, right? If you're not in the CNS, you're in the PNS, right? So they're out there in the peripheral nervous system. Whereas I would think of an oligodendrocyte as a squid. It's reaching out to multiple axons and making multiple segments of the myelin sheath on those axons. The Schwann cell reaches out to one axon and makes one segment. Okay? So it's like a one-to-one instead of a one-to-many situation. The other big difference is if you have damage to a Schwann cell, if you have damage to a peripheral nerve, you could actually have some recovery, right? And the reason for that is the chemical composition and this structural setup with Schwann cells will allow some regeneration, right? Some recovery if there's damage to a peripheral nerve. Centrally, we're not going to see that, okay? If you were to knock out one of these, you think about it, you knock out an oligodendrocyte, I mean, look at this. You're losing all kinds of points on that axon, on various axons. You're not just losing one, sort of one piece, right? How many of you have ever heard of MS? Yeah, okay, lots of folks, right? This is, and we'll talk in more detail later, this is a sort of an autoimmune, it's a degenerative disorder, right? What happens is you start to attack your Schwann cells, right? And you start to destroy that myelin sheath that's in your central nervous system, okay? If I were to ask you what's a symptom of MS, right? You could give me a variety of symptoms. The reason there are so many sort of different manifestations of that is it's which brain region is affected first, right? Individuals with MS often have difficulties with movement. Why? Because movement structures are going to be affected. They have difficulty with other nervous system function as well as central nervous system function, right? Depends on the nucleus that's been targeted in the brain. MS does not target Schwann cells because they have a different chemical composition. Okay, so they're not going to be affected by MS. Only those in the central nervous system are, which is unfortunately where you're not going to have any recovery if there's any kind of injury or neurodegeneration. So why did I tell you about Schwann cells then? So you could see it on this slide, not get too worried. Basically, they reach out and they create a jelly roll right around that axon. So there you go, right? Nice and protected. We're not going to let that signal leak out. We're going to let it travel through that axon fast. Okay. This is a cross section of that axon, right? We're just kind of chopping down. Okay. Questions about myelin? Nope. Great. Remember that time I told you about the blood-brain barrier? Now I'm going to show you a slide about it. Blood-brain barrier, we talked about this. There is one brain region, and this should have been like on a separate sort of point down here. How many of you have ever heard of the area postrema? No one? Okay, what does area postrema, it means it's the area at the back, right? How many of you have ever seen projectile vomiting? Oh, I know, right? That's so much fun. Typically we think about this with like kids, right? Like little kids will often ingest things that they shouldn't, and then they will right? They'll just shoot stuff out, right? It's another reason to avoid kids, right? So I think wherever there's a kid, I think about, well, how far can it vomit in any direction? And that's like you've got to stay out of the splash zone. So why am I telling you this story about projectile vomiting? There are a few places in your brain where the blood-brain barrier is weak, right? And one reason it's weak is because we need to know what's in your blood. Is it something that can cause you harm? And if it is, we need to try to get it out of your body. Where's the primary place that you put stuff into your body? It's your food hole, right? Okay, you eat things. You put things into your mouth. It goes in your digestive system. There are some other places you put things into your body, but that's the one we're going to think about, right? And if you put something in there, what's the easiest way to get it out? Well, let me take that back. What's the fastest way to get it out? Because the easiest way to get it out is just let it, go through its normal down and out process. But if you want it out quickly, a good old-fashioned vomit's the way to do it, right? So if it's in there, it gets into your blood, your brain goes, this idiot ate some poison. You got to get that out, right? So you're going to try to vomit that out. It's the quickest way to do it, okay? Does that make sense? Why do you vomit when you have like a viral infection? You're trying to get things out of your body, right? It's often why you also have diarrhea, right? So you just work both ends of it. Often, it's truth. So if you go to the doctor, often they will only give you medication to treat one of those outspouts, right? And they usually will give you an anti-nausea medication. They don't really care if you have diarrhea, right? As long as you stay hydrated. Because you still need an exit plan for that infection, right? Whatever that is. Vomiting's pretty rough on you, right? And so we could talk about all of the bad things that happen when you vomit. Bursting blood vessels in your eyes, rotting out your teeth, your breath is awful. If you have long hair, that's a hazard, right? That's why I keep my hair so short. Actually, I don't think I've vomited in like 25 years, so I got a good streak going. It's in the parking lot of a grocery store. I know, I remember exactly. I was like 8. It's close enough. Other questions about the blood-brain barrier, vomiting, circumventricular organs. You don't know what that is. That's these weak spots in the blood-brain barrier. We'll talk about those when we talk about ingestive behavior in a few weeks. Okay, so we're going to come right back to that. There's a picture of your blood-brain barrier. This is typically what a capillary looks like if it's out in your body somewhere. You got these nice gaps. Why do you have gaps? You want stuff to come in and out, right? Sometimes big things, okay? I'm not talking about just like little oxygens or carbon dioxide. I'm talking like big pieces of stuff, right? You can move some sizable items in and out. Notice how tight these cells fit together in the capillaries of the brain, right? So they're in there. They're tightly joined together. We're not going to let things come in and out, right? We're going to keep that sealed off. Again, unless it's something that is lipid soluble, right? If it's lipid soluble, so if it's like alcohol, this goes right there to your brain. All right. Types of neurons. We got all kinds of types of neurons. These neurons have different kinds of shapes. Is that important? Oh, absolutely. Do you need to memorize? You don't. Just know that if it's starting somewhere and ending somewhere else, if it's doing a slightly different kind of job, if it's collecting a different signal or a different type of signal from the outside world, it's going to have a slightly different shape, right? It's going to have some kind of different structure, right? How many of you have a car? How many of you have seen a car, right? Okay. How many of you could draw a picture of a car, right? There are all kinds of different vehicles, right? Some people have station wagons. Some people have minivans. These are all people we worry about because they have too many people they have to drive around. Some people have trucks. Some people have motorcycles, right? They're all doing the same basic job. They're taking you and maybe some other stuff from one place to another, but they're structured differently depending on exactly the nature of that job. Neurons are the same way. They're all doing the same basic thing, right? They're moving an electrical signal from one place to another, right? But how they're getting that electrical signal and where they're sending that electrical signal might be different, so they might need a slightly different structure. Do we want to talk about reflexes? I don't know. Maybe we do. By the way, this is the exact right way to determine if your iron is ready to use to iron your clothes. Just poke it with your finger. You'll know. If your finger gets stuck, it's ready to iron. That's how you know. I would like to say I don't know who would do this, and then I realize like half of you probably have done this, right? Anybody tried to iron their shirt while they're wearing it? Yeah, that was, thanks for being honest. Yeah, that's a bad idea, trust me. Look at some weird marks on your body. So, reflexes are important, right? We'll talk about reflexes a little bit. The problem is there are a lot of reflexes, in this one in particular, never involves the brain, right? Okay. How many of you have ever been to the doctor and they whack you in the knee and then you kick them in the face, right? Okay, they check that patellar reflex, right? Is there any way to prevent that from, have you ever tried to prevent it from happening just to make your doctor think you've got a neurological issue? Nobody's done that. You should try next time. You know what you'll do? You'll kind of like flop out of your seat like a fish, right? There's not really a way to prevent that. It's going to happen, right? You can try to dampen it a little bit, but it's a reflex. You're not in control of it. It's never going to your brain. There's not an inhibitory circuit there to stop it. It's going to happen. It's not the case with all reflexes. Some reflexes do involve the brain, right? And they can send signals down and they can inhibit that, right? How many of you, this is a great example, you're carrying something hot. What do you do if you're carrying something too hot? You should just drop it instantly, right there. Just chuck it away from you towards someone. You're not going to do that, right? You pick up something and it's hot and you want to drop it. You might try to inhibit that, all right? And so you can carry that somewhere safe so you're not injuring yourself and others. So some reflexes have some inhibition, some don't. We want to sort of think about that, okay? I just want you to look at this slide for 30 seconds. I don't want you to think about what's on it. Just stare at it. We're just going to leave it there for a moment. Okay? Try to let your brains catch up to your bodies. Right? Because what we're getting into next is, for many of you, going to be something sort of new. Because nothing I've said so far has been new, right? No. I think it has for some people, right? Okay, that's fair. This is going to be a slightly different way of thinking about sort of everything. How many of you are electricians? Nobody, right? That's good. But we're going to try to think about electrical signals. We're going to use words like potential, right? Membrane potential. These are sort of abstract concepts. I want to try to explain them in a way that you can have some understanding of what's going on, why they change, what happens when they change, and how this fits in to that whole story I told you, like on that first slide that had the neuron on it, right? And we talked about all those ion channels opening and closing, right? We're going right back kind of to that story, but we're going to zoom in on a very small segment. Okay. So let's think about a few things that we might want to do here. One, we do want to measure what's called the electrical potential of an axon, right? We've already determined from that great guy, Johann Mueller, that what's traveling through here, that's what's traveling through these axons, is an electrical signal, right? We know that. Great. Okay. But we want to measure the exact nature of that electrical signal, right? We want to measure how fast it's moving, peaks and troughs. We want to get the shape of that electrical signal, right? We want to know what's going on because that's going to be exciting. How might we do this? We get something called a microelectrode. And what is a microelectrode? It's a really thin, sharp piece of metal that you're going to jab right into an axon. Okay? Stick it into an axon, you can stick it into a neuron. It's going to give you some sort of reading on that electrical activity, right? How many of you have ever used like a multimeter or a voltmeter? You guess what I'm talking about? It's that usually they're like kind of yellow and they have a dial on them, and then like a red and a black wire comes out, and you just like poke stuff, right, until the dial changes, okay? It's a similar idea to that, but a lot smaller, right? So they're going to be very, very small. We can put one in the cell, and typically we need to put one outside of the cell somewhere, right? Because what's important is what's the difference between the inside of the cell and the outside of the cell. Makes a lot of sense, right? If I'm just measuring the inside and the inside, then I don't know, right? Is that, what information does that give me, okay? Let's think about this room for a moment. How well does the HVAC system work in this building? Well, if I don't know the temperature outside, I don't really have any idea about how well it works here on the inside, right? I can tell you the temperature inside. Doesn't matter what the temperature here is inside, right? It's important, what's that temperature relative to the outside temperature? What's that difference, right? Maybe it's 70 degrees in here. Maybe it's 70 degrees outside too. That doesn't tell me anything, right? But if it's 112 degrees outside and it's 63 degrees in here, then I can say we got a pretty good air conditioner, right? That's working pretty well, okay? We need to know that difference. All right, so what we're going to do is measure something called the membrane potential, okay? And there are different times when we might measure this membrane potential, but overall this is going to be the electrical charge across the cell membrane. So basically what we're saying is how positive or negative is the inside of the cell relative to the outside of the cell, okay? To make things easy for you, we have decided the outside of the cell is always 0, okay? It's always 0. Why is it always 0? Somebody said so. It was a good idea. We just kept doing it, okay? The outside of the cell equals 0 millivolts. Okay. If the outside is 0, then the inside, as it goes up and down, positive or negative, we can determine how far is that off from 0, right? We've got a reference point, okay? So it would be like we're measuring the temperature in the room relative to the temperature outside. And we're going to assume the temperature outside is always 0, right? And if the inside here is colder or hotter, then that's going to be, a negative or a positive. Right? We're not going to give you like an absolute number. We're not going to say, hey, it's 75 degrees. And here we're going to say, well, it's 80 outside, 75 here, so it's negative 5 degrees. That's going to be the answer we give you, right? That's what we're kind of doing here with this, this membrane potential. Okay? Does that make sense to everybody? It's the difference that's important. I sort of hate the concept of resting membrane potential. And the reason I do is because it gives you some idea that at some point your neuron is just taking a break. And it's not. Okay? But what we assume is that on average, if your neuron is not firing an action potential, if it's not really excited about something and sending that signal forward, then it's got to be at some sort of average voltage below that, right? Some kind of average voltage is just going to cruise along. I think in your book, does it use, anybody know, does it use negative 65 or negative 70? I can never remember. It doesn't largely matter. Let's assume it's negative 70 millivolts. It's going to be the resting membrane potential, okay? This is where your neuron would go to based on tons of things, electrochemical gradients, sort of the balance of ions and so forth. It's going to get you to this negative 70 millivolts, okay? In reality, you're not going to stay at negative 70 millivolts. The reason is those 10,000 contacts, right? Which are constantly telling you, here's an extra positive ion, here's an extra negative ion, constantly moving you back and forth, up and down, right? Close, but never probably resting on negative 70 flat line. You're always kind of just going up and down, right? Always up and down, until the point you get to that action potential. We'll talk about that in a moment. There are a couple words that we will use. These are probably new words for you, right? How many of you have heard of something that's polarizing? Yeah, like there are a lot of polarizing things, right? Okay. When we're thinking about polarizing things in this class, we're thinking about actual electrical currents, right? Movement of ions, okay? If we say something is depolarized or depolarizing, we mean that it's reducing that potential, right? And by reducing the potential in this case, because we're always hanging out at -70, to reduce that we have to get closer to 0, right? We have to continue to sort of increase the positive ions that are inside that cell, right? So we're going to move towards 0, okay? If we are hyperpolarizing, it means we're making it more polarized. So we're going to move farther away from 0. In this case, we're going to make something more negative, right? So if we're typically at -70 millivolts, we'll just kind of assume that for now. If we want to hyperpolarize, we might go to -80 millivolts, right? -85 millivolts. Those are movements, those are hyperpolarizations, movements away from 0, making that difference between the inside and the outside of the cell larger, okay? If we depolarize something, then we go from -70 to -60 to -50, right? Because we're moving closer and closer to 0, we're making that smaller and smaller, that gap, that difference. Does that make sense? All right, awesome. All right, what are some things we want to think about to move ions? One of those is going to be diffusion, right? You guys familiar with diffusion? If you have a high concentration of something and it's able to move, it will move to an area of lower concentration so you can equal out those concentrations. That's a pretty basic force. There's also what we call electrostatic pressure. Okay, so we want to think about the charges here. Because we're dealing with ions, anybody know what ions are? There's charged atoms, right? So we're thinking about things like potassium, sodium, chloride, and then there's something we call an organic anion. Don't worry too much about that. We'll talk about that when we need to. But we're talking about things that have a charge, right? They have a positive or a negative. In these cases, we're typically thinking about things that are single, but there can be things with charges that are more than one atom, right? How many of you remember all those amino acids? Yeah, some of those had charges, right? Somewhere around half of them have charges, positives or negatives, right? And so those things can push or pull other positives and negatives kind of depending on what's going on. That's awesome because you create all those alpha helices, right? Those beta pleated sheets. That makes really cool shapes and structures for your proteins. For our purposes, we want to think about pushing things apart, right? So if we get a bunch of potassium and a bunch of sodium together, they're going to push each other, right? Because they're positives, right? You get a bunch of positives together, they're going to start repelling each other. We also have something called the sodium-potassium transporter, right? How many of you have heard of the sodium-potassium transporter? It's an awesome thing, also known as the sodium-potassium pump. Or the sodium-potassium ATPase, right? Let's be capital P. We should keep in mind anything that, for our purposes, that ends in ASE, that's an enzyme that chops stuff, right? It's pretty simple, okay? So if it's a whatever ACE, it's chopping something, whatever that is, and when you chop a molecule, you release some chemical energy, right? ATP works that way. ATP being that sort of energy currency, right? When you take ATP, you chop off a phosphate, what do you get? Well, one, you get ADP, right? That's di instead of tri. And you also get some chemical energy that's released. You can convert that into sort of some mechanical energy, right? So the sodium-potassium transporter is constantly swapping sodium and potassium, right? It's pumping out sodium, pumping in potassium, right? Because we need to set up this concentration gradient for an action potential later, okay? We'll talk in some details about that sodium-potassium pump in a bit. Intracellular fluid, this is inside the cell, You have high levels of the organic anions and high levels of potassium. Okay, that's what you're typically holding on to inside that cell. Okay. Outside the cell is mostly, it's just salt water, right? All right, salt water. We have sodium and we have chloride, and a lot of it. How much of it do you have? As much of it as you need. Okay? We don't need to think about running out of sodium. We don't need to think about running out of chloride, because we are really just, our brain cells are just sitting in a vat of salt water. Okay? So we don't need to think about running out of either of those ions. Okay? Sort of an unlimited supply of those. All right, so here we go. This is outside the cell. Here's inside the cell. Remember, high concentrations, sodium and chloride. Inside the cell, high concentrations of potassium. And what's interesting about that organic anion is it cannot leave the cell. It's so big, it's kind of bulky. There's not a special channel for it. It's not lipid soluble, so it's not going to slide right through there, right? Things with a charge are not lipid soluble. If it carries a positive or a negative, it has to have a special channel to get in or out of the cell. Okay? It's not just going to freely move in and out if it has a charge. Okay? That's what's awesome, because we have ion channels that are going to open and close and allow things like sodium, chloride, potassium to move. There's also another guy on here, right? How many of you guys have heard of calcium? Okay. You also have calcium ion channels, right? So you need to move calcium. Okay. There are a few places where we'll think about calcium channels being important. One is going to be learning and memory. Anybody else know where there might be some calcium channels that's important for you? What? I thought somebody said heart. Did anybody say that? Go ahead and raise your hand just to take credit for it. Yeah, it was a great answer. Great answer. Even though it sounded like it came from over here in a female, I'm going to give you credit for it. So your heart uses some calcium channels. This is why individuals who have sort of genetic mutations that alter their calcium channel function typically don't live. It's a non-viable mutation because it alters cardiovascular function in such a way that your heart's not going to work, you're not going to exist. Okay? There are things called channelopathies, right? So these are diseases, disorders, however you want to think about it, that the structure of the ion channel is altered in such a way that it doesn't function properly. Some of those are fine. You can just kind of get around like a normal person, no worries. Again, some of those are going to be sort of non-viable, right? Particularly if they're involving those calcium channels. All right. Here's that awesome sodium-potassium pump we just talked about. You're going to pump out 3 sodium, pump in 2 potassium, right? Every time you pump out 3 positives and only 2 positives in, you get a net gain of 1 negative on the inside, right? Remember that time I told you about resting membrane potential and what I say that was? Negative 70 millivolts, right? So if we do something to change that, right? And all of a sudden, let's say now our membrane potential is positive 40. What do we have to do? We have to work to get back to negative 70 so we can reset that membrane potential for another action potential, right? The sodium-potassium pump is going to be part of that mechanism, okay? Part of that reset mechanism, moving these ions in and out. This is a huge consumer of energy. That's why we also call it the sodium-potassium ATPase, right? I just talked to you guys about that. It's taking a lot of ATP, chopping off a phosphate, using that to sort of operate this mechanical pump to get those ions moving from one side to the other, okay? Why do we need special channels for ions to move? They're charged and they're not lipid soluble, right? So if this ion tries to get through here, it's not going to work, okay? It's not going to fit. Does that make sense? All right. What do we know about action potentials? Well, I've sort of just skirted around the action potential. I'm going to keep doing that for a little bit, right? At some point, we're actually going to talk about the action potential proper. We'll get there in a few moments. But the whole point of this process is to fire an action potential, right? Because the action potential is the signal that's going from one place to another. So what happens? The cell becomes depolarized. What does that mean? That neuron goes from -70 to something less than -70, right? It moves up a little bit closer, becomes more positive. It doesn't go on the other side of 0. I want to keep that in mind right now. It just gets slightly less negative, okay? When it gets slightly less negative, it reaches what's called the threshold of excitation. That sounds exciting, doesn't it? Maybe I should just say the threshold of excitation, right? Really put some emphasis in there for you. So this is important. When you reach that threshold of excitation, it's like the top of a roller coaster. You guys ever been on a roller coaster? So when you get to the top, like all the way up, you could have a complete fit and they'll let you off, right? Okay? They will stop the roller coaster. You could, I don't know, projectile vomit. And they will stop and they will get you out, right? But once you get to the top and they let you go, you got to hold on until it stops, right? There's no way. You got to keep going, okay? And that's exactly what the threshold of excitation is. You get up to that threshold of excitation, and action potential is going to happen. There is no way to avoid it at that point, okay? This is mostly because we're moving around a bunch of sodium and potassium ions, right? We're going to move those sodium ions in. Okay, we're going to flood in a bunch of sodium ions, and then we're going to leak out a few potassium ions because later we're going to need that concentration gradient difference to get reset back to that resting membrane potential. Okay? Any questions about this? We're going to talk about it again in a few minutes. Okay. Should probably talk a little bit about ion channels, right? And yes, they all do look like bell peppers. It's not really true, but ion channels, right? They are exactly what they say they are. It's a channel, it's a tunnel, it's a hole in the membrane of your neuron created by this protein structure that allows a specific ion to go through it, right? Okay. How do we know that only one type of ion can go through? Well, the first thing you might think about is size, right? Ions have different sizes, okay? So the size of the opening is important. What's also important here is the lining of that ion channel, right? Because maybe, like if you make the channel so big that a bigger ion can fit through, then you'll think, well, all the smaller ions will fit through there too, right? Well, we got you covered there. So remember those amino acids I told you about that all have different charges sometimes, right? We can line this channel with a different sequence of charges that can prevent certain ions from coming through. Even if they're small enough to fit through the channel, the charge might not match in the right way, so they're not going to allow that ion to slip through, okay? It's pretty cool, actually. So what ion could this be? Well, if this is the inside of the cell and this is the outside of the cell, and the outside of the cell, what are your options for ions typically? Sodium, could be chloride, right? Okay. Does that work? It's a great picture, right? Looks like a bunch of radishes next to a bell pepper. You just imagine like when you were a sophomore in high school and you were like, make a model of the phospholipid bilayer with ion channels, right? And you just go like, I don't know, I'm thinking about like skewers and radishes. I don't know. You should try that, Kyle. You should throw a party, and you should make all of the food look like... No? I don't think that's going to be popular? You don't think it'll turn out well? This is great for parties like Super Bowl parties, right? And people can just come along and like pluck off a roasted radish when they want, a piece of a bell pepper. I think it's a great dish. It's on my Pinterest page. I don't even know what you can put on it. Can you put that on? I don't know what Pinterest. I know it exists. Does that sound like something? Can be. It is now. If I were to tell you, like, there's only one slide the entire year, the entire semester, that I want you to understand, it would be this one. This is probably the most important slide in this entire course. That doesn't mean ignore all the other slides. That just means pay a slight bit more attention to this one. Okay? So what is this slide? This slide is telling you what is happening during an action potential. Right? And why is an action potential so important? Because everything that you think, everything that you do, every calculation, every moment, every breath, every time your heart beats, we're thinking about action potentials doing that, right? Action potentials are controlling that process. Okay? You're simply just a collection of action potentials housed in phospholipid bilayers. Right? That's it. It's not very significant at all. I don't know, I think it's exciting. Takes a lot of responsibility off of you, right? Next time you screw up, you're like, man, I'm just action potentials. What'd you expect? So, what's happening here? All right. This number, that's the resting membrane potential, right? That's where we're hanging out. Now, in reality, is it just a flat line, negative 70? No, because we're getting those yes and no votes, right? We're getting ion channels that open up for sodium and some that open up for chloride, right? So in reality, we're kind of cruising along doing this, right? Just up and down a little bit. Okay? At some point, enough positive ions come into that cell to push that membrane potential up enough, right, to hit that threshold of excitation. It's not very far. So if we look at this scale, down here we have negative 70. Here we have 0, right? Here we have 40. So if you break that up into kind of 10, 20, 30, 40, you're thinking that's really only about a 10 millivolt difference, right? From normal resting membrane potential up to the threshold of excitation. Okay. We don't want that difference to be too far. The farther you raise, right, that threshold of excitation, the harder it is to fire an action potential, right? The harder it is to fire an action potential, the more difficult it is to avoid being eaten by a bear. Okay? So you want your threshold of excitation fairly low. Okay? You don't want it too low, because then what happens? Your neurons are constantly firing, right? And if neurons are constantly firing, guess what happens? You have no idea about what's going on in the world, right? There's too much noise, okay? And we don't mean noise like you're hearing sounds, although that could be happening. There's too much irrelevant information coming in. There are too many accidental action potentials. You have accidental action potentials anyway, right? You're kind of sitting at this level, just ready to kind of move up and get ready to go, right? Sometimes you're going to accidentally fire an action potential. Happens all the time. We've got an algorithm to cancel those out, so it doesn't typically cause us a problem, okay? But if we drop the threshold of excitation too far down, then we fire too many accidental action potentials, and we don't know what's real and what's not real, okay? So that's kind of cool, right? So here we go. We've reached that threshold of excitation. We have opened sodium channels, and sodium is rushing in. And by rushing in, I mean it's moving in just as little as possible. It's not actually like coming all the way into the middle of the soma and like bouncing around the nucleus, right? It's barely crossing in, just getting to the other side of the membrane, right? The reason we don't want the sodium to go too far in, right, is the farther the sodium goes in, the harder it is to get it back out, right? And we need that sodium back out so we can fire another action potential, right? We want to fire another action potential because we want to try to use that neuron more than once, right? It's going to keep firing action potentials. Anybody know how long an action potential takes? We're talking a couple milliseconds, right? Very, very short time, Okay, we're talking about a very, very short time here. We're going to fire one and reset. Fire one and reset so we can send another signal right behind it. Okay? And that combination, that timing, that pacing of those signals is what's going to tell us the information about what that cell is receiving, okay? What decisions it's making. So we open those ion channels. We allow the sodium to come in just a little bit. What happens to the membrane potential? Because we're allowing in positive ions, right? The membrane potential becomes depolarized, and it continues to depolarize. And it will continue all the way up, and it'll peak right around positive 40 millivolts. During this time, right, we have started to open some potassium channels and allow the potassium to leak out from the inside of the cell. Okay? How does this happen? Sodium was in a high concentration outside. We opened a door to allow sodium to come in. When sodium comes in, it's all rushing in because we have that high concentration outside. Remember I said we're never going to run out of sodium on the outside because it's just a big vat of salt water. That's creating some electrostatic pressure, right? Because we have all that positivity coming in. When we open the potassium channels, okay, that's an easy escape route for some positive ions. So the potassium will go out through the potassium channels. Sodium can't go out that channel. Why? Because it's not potassium, right? It's designed specifically for the potassium and not the sodium. All right. When we reach the peak here, what's happened is the sodium channels have become refractory and they've closed. Okay? We can't open them anymore. There are two sorts of refractory periods that we want to think about here. The sodium channels, there's what we call the absolute refractory period and the relative. Okay? When we're talking about the absolute refractory period, this is a time when the sodium channel cannot open no matter how much you try to stimulate that channel to open. Okay, it's going to be impossible. It's not going to open. The relative refractory period is a time period after that where it's more difficult to open that sodium channel, but it's not impossible. Okay, with the right amount of motivation, with the right amount of stimulation, it would open, right? But it's going to be more than what's normal. Okay, it's going to require a little extra push to get that opened. This is going to become important here in a few moments when we think about the direction of an action potential, right? How many of you love action potentials that go halfway down and then turn around? Nobody likes those. Because you know what happens then? You die, right? You go, hey, I should tell my heart to beat. Halfway down, the signal comes back up. That's a bad idea, right? It needs to go all the way down, okay? So we've reached the peak of the depolarization here. Sodium channels have closed. We still have potassium leaking out of the cell. Because we're still letting positive ions go out, guess what happens to the membrane potential? It starts to become negative, right? Because we're still leaking out positive ions, okay? So we start to drop back down. Eventually those potassium channels will close, and then that sodium-potassium pump will work to bring you back to that resting membrane potential right around negative 70 millivolts. Okay? Questions about that? I will guarantee you, because I'm going to make the exam, so I know what's going to be on there. I'm going to ask you in detail to tell me about an action potential. Okay? What I would like to see you tell me is the information we covered for this slide, right? See, that's why I told you it was so important, right? Yeah. Not trying to trick you there. I'm just trying to be transparent, let you know this is an important slide. That was important content. I told you ahead of time. I'll tell you right after it. That way you know. Circle it, highlight it, rip that page out of your book, throw the rest of the book away. Whatever it is you need to do so that this is the content that you study, okay? This is going to be important. I'll remember I told them that. Thanks. I might forget. All right, so action potentials, right? There are a couple things we should talk about here. Should take just a moment. I got a notification from Blackboard. I'm trying to figure out who just took a quiz.
Did they pass?
They might have passed that one, but they should have been listening to the action potential story, because they might fail that. The problem is with Blackboard, I'll get a notification like 12 hours after you do something, so it's in no way helpful. So it's quite possible no one is doing anything on Blackboard. How many of you love giant squids? Yeah, there's nothing in the book about giant squids. You'll think it's about giant squids. So what they will do is they will, how many of you love the Oxford comma? Losers. I hate the Oxford comma. You know why? You should have just said your sentence the right way the first time, and you wouldn't have to put extra stuff in there so I know what you're talking about, right? If you just phrase it correctly, you don't need extra punctuation. Trust me, I know this. And the book's the same way. Every time you read a book about Hodgkin and Huxley, man, I remember that Huxley name for some reason, right? I think I told you to remember that last name, right? Okay. Hodgkin and Huxley, a couple guys, bright guys, they decided to record an action potential, and they will tell you every single time in the book from the giant squid axon. And then they will take a paragraph to tell you it's not the axon of the giant squid, it's the giant axon of the squid. Right? And so why they didn't tell you in the first place, Oxford comma folks, that it was just squids have giant axons, so that's why they used the squid. It's got a big axon, right? Because it's got a big axon, it's easy to jab with electrodes, okay? That's the only reason they did their experiments in squid axons is because they're giant axons. Right there, that's the story. It wasn't a giant squid, it was a regular squid, right? It just has giant axons. They should tell you that. In the beginning, it's just a frustrating point for me. So, Hodgkin and Huxley grabbed a couple microelectrodes, grabbed a couple squids. They're like, watch this, okay? And they would send a signal from one electrode to the other electrode in that squid axon, and they can measure things, and we learned a few things from that. One of those things is there's something called the all or none law, okay? Here's the deal. You're going to fire an action potential. If it's an action potential, it's going to look just like every other action potential. They're identical. You're either going to fire it or you're not. There's no half action potential, right? Okay? It's a full action potential. And it looks just like this. That's it. We also know that action potentials travel in one direction, right? Once you start an action potential in one place, it's going to travel down that axon somewhere else. Now, why is that? This is because of the refractory period of the sodium channels. Once those sodium channels open and they close, they can't open up again for a bit, so that action potential has no choice but to go forward. It can't go backward because it can't reopen those sodium channels because they've closed. So that creates a gap, right? Creates a gap between the peaks of the action potentials so that you can determine that they're individual spikes, right? That they're individual action potentials. Okay? Does that make sense? We also learned this thing called rate law. Don't have to worry too much about that. Basically, a stronger stimulus is going to create more action potentials. Here's that propagation of the action potential. We do call this saltatory conduction. It has nothing to do with salt. It has to do with leaping, right? And so the action potential sort of gets that turbo boost from one node of Ranvier to the next through those, well, it goes through the axons and then gets the boost there at the gap in the myelin sheath. All right. We have to send the signal. I mean, so far we've just been inside one cell, right? We've not really thought about the way that we're interacting with another cell. We're just stuck right in there. We really need to try to get this information from one cell to another, right? We have to communicate between, not just within ourselves, right? So we got to send that signal out. How many of you have really great ideas? I doubt it, but thanks anyway. A few hands went up. But if you have a really great idea, that's awesome, right? But how do you convey that idea to someone else? How do you communicate that idea, right? How do you get that out to other people? Okay, that's what we need to think about here. This neuron's got a great idea. It's an action potential. I'm sending that action potential down my axon, right? How am I getting that to another cell so that I can affect the activity, the actions of that other cell, right? That's what we want to think about now. All right, we call this synaptic transmission. Why do we call it synaptic transmission? Well, we call the space between the neurons their synapses, right? And so we're sending that signal across that synapse. Okay, we've got to get that message from one cell to the other. We have a couple labels here we use, presynaptic and postsynaptic. Just means the cell before the synapse and then the cell after. The cell sending the signal, the cell receiving the signal. It's not so bad, right? Between most of your brain cells, right? The reason we put a gap between most of them is if one gets activated, we don't want all of the rest of them activated at the same time. If all of your cells were connected directly, right? If there was an electrical change in one cell, that's going to cause an electrical change in all of the other cells at the same time, okay? So imagine you go into your house and you've got one light switch. And when you turn that light switch on, every electrical appliance you own turns on. Television, the washing machine, the disposal, whatever it is, it turns on. What if you want to turn it off? You got to turn everything off. That seems like a bad idea, right? That's a bad way to wire your house. Okay, you want individual switches so you can turn things on and off as you need to. Your brain has gaps between the cells, right? So you can activate different cells in different circuits at different times. We can send these signals differently, right? It gives us a little extra control, okay? So we take that electrical signal, we turn it into a chemical signal, that's the neurotransmitter, and we turn it back into an electrical signal. Seems like a lot of work, seems like a lot of effort, but that gives us some very detailed and tight control over that signal and how we send that signal forward, right? Okay. If we're talking about postsynaptic potential, that's the changes, either hyperpolarization or depolarization, on the other side, right, when we open a few ion channels, and what's that do to that membrane potential, okay? Here's one that's really going to confuse you. A binding site, right? This is where something binds. It's the site where it binds. I know it's a very complicated definition. Just want to make sure you have it. Basically, you've got a receptor, and there's a thing called a ligand. What is a ligand? Well, it's some kind of chemical thing, right? Don't worry too much about it. It's a fancy word. The ligand is going to bind onto the receptor. Where does it do that? At the binding site. It's not a big deal. We've already talked about dendritic spines. Okay, we do want to take a moment here and think about another concept. So this is the second time today I'm going to give you what I like to call keys to transcendental enlightenment. Right? The first was lipid solubility, and that puts you well on the path, right, to being a fully realized person. This next one is just going to get you even closer, right? And that is surface area to volume ratio, right? If you understand the importance of surface area to volume ratios and how that affects biological systems and the structure of biological systems, you can take this concept, you can apply it to your digestive tract, right? How many of you know about the way the structure of cells, I know you weren't ready for that, in the intestine, right? They have all of these little fingers that stick out, right, on those cells. Why? It increases surface area relative to volume so that you can increase the surface area for absorption of nutrients, right? Why is your cortex all crinkled up? If you'll remember, last week we talked about how big your heads are and how small your mom's hips are, right? And how you're not going to fit out. If you were to take your cortex and spread that out, that thing's like 3 feet by 3 feet, right? Okay? Can you imagine trying to fit a 3 foot by 3 foot cube out the birth canal, it doesn't work, does it? Okay, so we're going to crumple it up. It takes up less volume, but we can increase surface area there, right? We're trying to do the same thing with these dendritic spines. Every time we add a little spine to a dendrite, we're increasing the surface area. We're increasing a place where we can have a contact, one of those 10,000 contacts, right, without making the cell too large. Okay? Surface area to volume ratios is exactly why you have donut-shaped red blood cells and not spherical red blood cells, right? If you had spherical red blood cells and you put oxygen in the middle, you know how long it'd take for that oxygen to get out of that cell? It'd take too long, right? You're blasting past the place that needs it. When it's donut-shaped, right, reduces the distance from the center of the cell, wherever you might have that oxygen, to the surface so it can jump off, and it increases the surface area so you can load and unload more oxygen at any given time. It's a very broad concept, right? So I think it's going to help you here if you start thinking about that in terms of dendritic spines, global cortical arrangement. I mean, there's a bunch of stuff here for surface area to volume ratios. All right, presynaptic membrane, postsynaptic, we talked about that, not a big deal. Well, here's a synapse. Not too big of a deal. We've got our terminal button coming down. Okay, it's going to contact on a dendrite somewhere, on a cell body. It can contact on an axon. It can contact on another terminal. We've got a lot of options here. Sort of depends on what we want to do, what kind of influence we want to have on that postsynaptic cell, where we would want to contact that if we're a synapse. Okay. So what happens here? That action potential travels down this axon. Okay? I'm not going to expect you to understand too much of what happens here, right? But that electrical signal basically starts a series of events that takes these synaptic vesicles. And what's a synaptic vesicle? It's simply a sphere of phospholipid bilayer, and tucked inside of that is typically neurotransmitter molecules, right? So it's basically, you think about water balloons, it's like a water balloon filled with dopamine. Sounds pretty exciting, right? And they're all sort of docked, right, in this terminal. They're all just sitting there waiting on that electrical signal to come down. There's some other stuff involved, but they undock and they float their way down to the end of that membrane. When they reach that exterior surface, right, they're on the inside, but when they reach that membrane, they merge, and as they merge, they start to open up, and they dump out all of their contents into what we call the synaptic cleft, which is this space here between the presynaptic cell and the postsynaptic cell. Okay, so they're going to dump out dopamine, they're going to dump out acetylcholine, they're going to dump out serotonin. Whatever neurotransmitter it is that they have stored in there, or a combination of neurotransmitters they have stored in there, they're going to dump that out. Okay, it's that simple. Once they dump out that neurotransmitter, those neurotransmitters, through the magic of diffusion, right, find their way over to that other membrane, right, that postsynaptic membrane, and what do? They bind at the binding site, right, of those receptors. What happens then, there are a couple of things that can happen. End result is we're going to open an ion channel. Okay, what ion channel are we opening? Well, whatever one has, you know, whatever one's activated by that ligand, right? It could be any ion channel, really. Typically, we're thinking about sodium or chloride. We'll add calcium into that group as well. We open that ion channel. Ions come in. Guess what happens then? Well, we might get closer to that threshold of excitation, right? We're working our way back to another action potential, right? Because we've adjusted that membrane potential a little bit. If enough of it's happened in the right direction, we fire an action potential. That action potential travels down the axon, right? It makes it to that next terminal. When that signal reaches that terminal, causes the undocking of those synaptic vesicles, right? You've heard this story. Okay, it's the same thing, right? It just happens in the next cell. We release those molecules of neurotransmitter into that synaptic cleft, and we can open up another set of ion channels because of that binding site business and all that fun stuff, and it just keeps going, right? That's so exciting, right? You guys seem thrilled. Questions about that? No? Okay. Let's think about different kinds of receptors. Okay? There are two broad classes of receptors that we want to think about. Ionotropic, and we'll think about metabotropic here in a moment. Ionotropic receptors, right? These are guys that are directly associated with an ion channel, okay? Directly associated with an ion channel. So some neurotransmitter, whatever that is, and then Kaylin, it goes to that binding site, right? It plugs into that binding site. There's that neurotransmitter. Plugs into the binding site. When it does that, it causes a structural shift in the shape of that channel, right? So when that ligand binds, it actually starts forcing, think about those amino acids again with those different positive and negative charges, right? It starts forcing those to move, and it opens that channel. And it allows ions to flow through from the point of high concentration to the low concentration, right? So let's imagine we could tell you a specific story. This could be a sodium channel, so these could be sodium ions. This neurotransmitter here, let's just say it's glutamate. I mean, it could be dopamine, it could be whatever you want it to be. It could be serotonin, acetylcholine. Okay, it's like 50 options. Okay, it could be any of those. That molecule binds, it opens the channel, sodium ions rush into the cell, and you fired an action potential. It's not so bad, right? Ionotropic receptors, direct. Okay? The ion channel is literally right there with the receptor. Okay? They're sort of one and the same. They're integrated into one structure. It's direct. It's a one-to-one relationship. You've got one molecule of neurotransmitter, you open one channel. Okay? So it's direct. It's one-to-one. It's very rapid, right? There's no time delay. As soon as there's binding, it opens, ions come through. It's that simple. Okay? We're going to contrast this with these metabotropic receptors. These are a little different. How many of you have heard of like second messenger systems? G protein coupled receptors, right? That's the business we're dealing with here. You still have a binding site. You still have that receptor. You still have some molecule, whatever molecule that is, right? Let's think it's acetylcholine. That sounds like a fun one. Acetylcholine comes in, it binds to that receptor. What does that receptor do? It sends out that second messenger to other places that then opens ion channels, okay? So this is not direct, right? The ion channel and the receptor are in two separate locations, right? They're geographically separated. They're not integrated into one structure. So it's not direct. It's not a one-to-one relationship, it's a one-to-many, right? When you activate one receptor, you can actually open multiple ion channels, okay? We call this a cascade, okay? You guys familiar with cascades? I don't mean the stuff you put in your dishwasher, right? So a cascade is something where you have one, and then the next thing you know, you have three, and like each of those three has like three. This is like a cat you didn't spay or neuter. That's a cascade, right? Because at first you only had one or two cats, and then how many cats do you have? 50. It happens, right? And it's much easier to get rid of 2 cats than it is 50. In fact, it's much easier to get rid of parts of 2 cats than it is 50 cats, right? Because there are a few parts you can get rid of so you don't have more cats. We'll talk about that. It's like Chapter 10, reproductive behavior. We'll talk about what parts you can get rid of and not have more cats. Okay? Right, Kyle? We'll let the vet figure that out. Just take your cats in and say, I don't want more. The 2 I have are fine. All right, so we have that cascade. Because there's a geographical separation here, there's also a little bit of a time lag, right? So this takes a little bit longer. Now, we're still talking on the order of like milliseconds. We're not talking about hours, right? We're not even talking about minutes. We're probably not even talking about whole seconds here, right? But it takes a little bit longer than an ionotropic receptor because we do have to send that second messenger, right? And it has to travel some distance. All right, so we talked about synapses, we talked about vesicles, don't worry about release zones. That's also a very, very complicated definition. That's the zone where they're going to release the vesicles, right? So put that right up there with binding sites. Spend a lot of time studying that. Postsynaptic potentials, what do we want to think about here? Oh, let's go back up a slide. We do want to think about these words briefly. Excitatory postsynaptic potential. How do we cause an excitatory postsynaptic potential? Well, we open ion channels that allow in positive ions, right? When we think about, let's open up sodium channels. Okay, that makes sense. How do we cause an inhibitory postsynaptic potential? Well, we can open up negative ion channels, right? So negative ions are cations, right? Or I mean anions. So we could open up chloride channels, right? So more chloride comes in, and then we have an inhibitory, so we're less likely to fire an action potential. All right. At some point, you got to stop this, right? At some point, you have to say, you know what? This action potential business, this activation business, these neurotransmit, it's happened. It's gone on too long. We say we need to stop it. There are a couple ways you might do that. One is through the process of reuptake. Okay? You've released, let's say, a bunch of serotonin. Awesome. Serotonin is opening ion channels. But you've decided, okay, time's up, serotonin, come on back home. So what do you have? You have these, they're really essentially just arms, right? So they're just protein arms that reach out from the cell that released serotonin, and they suck that serotonin back in to that same cell, right? So reuptake, we're just pulling that serotonin back in. This is why things like selective serotonin reuptake inhibitors increase the availability of serotonin. If we stop or inhibit that reuptake process, we leave serotonin out at the synapse longer for it to do its action, okay? The other thing that we might do is called enzymatic deactivation. Okay? Instead of pulling that neurotransmitter back into the cell that released it, we might just go out and destroy that neurotransmitter, right? And we would do this with something for acetylcholine. We would use acetylcholine esterase. Hey, do you remember that time I told you anything that ends in ASE is a chopper? Right? It's just going to chop apart acetylcholine into two things that are not acetylcholine, and then they can get cleaned out of the way, right? And they're not going to have their effect anymore. Okay. If you didn't have acetylcholine esterase, guess what you would do? You would flex a muscle one time, and then you're done. You're stuck there. You're never going to release that, right? Okay, so if you're like, hey, and then you're stuck. That's all you got. So it's so great that you can release that and continue to move your arms. I think that's important to continue to move your arms, right? You have to really think for a long time, like, which movements do I want to make? And the problem is you're like born an idiot, right? Because babies have no idea. And you're going to make some like wailing cry, and then you're stuck. And then our species no longer exists. So that's why we have acetylcholine esterase. The continued existence of our species. Isn't that great, Kyle? We'll talk about stuff with acetylcholine later. Movements of ions, we talked about this, right? Not a big deal. Excitatory, postsynaptic potential. Inhibitory can actually, if the positive moves out or the negative moves in, not a big deal. What about reuptake? Okay, again, we're trying to grab whatever that neurotransmitter is and pull it back into the cell that released it. Just a diagram showing that. Don't worry about neural integration. Basically, this is just math, right? Your individual neuron is going like, hey, how many positive ion channels do I have open compared to how many negative ion channels do I have open? Which way are the ions moving, in or out, right? If they move enough positive in, then action potential. If they move too much negative out, well, I'm not going to do an action potential, right? What do we know about autoreceptors? This is actually kind of cool. How many of you have ever done something? Yeah, and then you needed some feedback about whether or not you did enough of it, right? That's what an autoreceptor is, okay? You release neurotransmitter, the autoreceptor is going to monitor that. Once the autoreceptor is activated, it's going to say, hey, you've released enough dopamine, I got the signal, stop releasing dopamine, okay? It's that simple. Later in a couple weeks, we'll talk about agonists and antagonists. We'll talk about how drugs can influence the release of neurotransmitters, and we'll talk about blocking autoreceptors, activating autoreceptors, fun things like that. That's why you need this chapter so you can understand chapter 4. Other types of synapses, basically you can make a synapse wherever you need to. Okay, so don't get caught up on it's got to be onto a dendrite, right? It can be onto anything. In fact, one of the most powerful places you could put a synapse is like on an axon, right? You could put like a real stop to things right there, like right there where the axon starts. Go, I don't think so. Let's just inhibit that action potential that was coming through, right, before it even starts. We will talk about hormones and other such things as we need to throughout the semester. Don't worry too much about it. We'll talk about the steroid hormones later. Questions? No questions, no comments, no concerns. Stop there.